Welcome to another session on biomedical instrumentation. In our previous sessions, we have understood the different systems in a human body and the development of biopotentials as a result of the functioning of the different systems. And if we can extract these biopotentials, they are capable of diagnosis and therapy of various diseases in the human body. We also understood the need for biomedical instrumentation in acquiring these signals. And in today's class, we will understand the methods for the acquiring of these signals. So, this presentation is about the electrodes and the electrode theory to acquire the biopotential signals. Now, bioelectric events are picked up from the surface of the body, and the devices that convert the ionic potentials into electronic potentials are called electrodes. Electrodes are nothing but electrical terminals or contact points for obtaining voltages from the surface of the skin. And in between the skin and the electrode, there is an application of electrolyte paste, or more commonly called as jelly, which are used to lower the skin impedance while acquiring the biopotentials. Now, to understand the theory of electrodes, let us look into the various interfaces that are found due to this electrode tissue interface. So we have the tissue or skin on one end, the electrode or the instrument on the other, and in between the electrolyte. Now, because of this, two interfaces are created. One is due to the electrode or the metal and the electrolyte, and the other is the electrolyte skin interface. Let us take up the first interface, which is the Metal-Electrolyte Interface. Now, let us assume that the electrode consists of metallic atoms C. The electrolyte is an aqueous solution with cations of that of C+ and anions of the aqueous solution. Now, transfer of charge between carriers represented by the chemical reaction is as shown in this clip. So we find that the cations can the metallic atoms can dissociate as cations and electrons, while the anions can convert into atoms and electrons. So N is the valence of the cations and M is the valence of anions. So the net current that crosses the interface, now this is the interface, the net current that crosses the interface from the electrode to electrolyte consists of three parts. So this is the total current. The first set of current is due to the electrons that is moving in opposite direction to that of the current in the electrode. The second one is due to the cations moving in the same direction as the current, and the third one is due to the anions, which is moving in the direction opposite to that of the current in the electrolyte. Now, as a result of which, a net charge gradient is created, which is nothing but the difference of potential at each electrolyte, and the spatial arrangement of this is called as the electrical double layer, as shown in the figure. The double layer is known to be present in the region which is immediately adjacent to the electrode and can be represented as two parallel sheets, here you find them, of charge of opposite sign separated by a thin film of dielectric. Therefore, the metal-electrolyte interface appears to be consisting of a voltage source in series with a parallel combination of a capacitance and a reaction resistance. The voltage that is thus developed is called as a half cell potential. So this is the representation of the electrode-electrolyte interface. The half cell potential is equal to the electrode potential of the metal, and all electrode potentials are measured with respect to a reference electrode, which is usually that of a hydrogen absorbed on platinum black. So the capacitance represents the charge developed at the phase boundary, whereas the resistance depends on the conditions associated with ion migration along with the phase boundaries and inside the diaphragm. So when the bioelectric events are recorded, the interference signals are produced by the potential differences of metal electrolyte and electrolyte skin interface. Normally, these potential differences are connected in opposition during the recording procedure. So this shows the equivalent circuit of a pair of electrodes for acquiring the biopotentials. All electrode potentials are measured with respect to a reference electrode, usually that of hydrogen absorbed on platinum black. This is an inconvenient electrode to make, and therefore other alternative electrodes which may have fairly stable and repeatable potentials are employed. So electrode potentials of some of the commonly used metals in the electrochemical series with respect to hydrogen is given in these examples. It is found that silver chloride electrodes have been found to give almost noise-free characteristics. They are also found to be acceptable from the point of view of long-term drift. So let us now observe the chemical reactions with respect to silver chloride electrode. So this is a pair of electrodes for measuring the biopotentials or a bioelectric event, and there is the Helmholtz double layer created, and the first charge difference is represented by the half cell potential, and later on the next level of potential gradient is represented by the parallel combination of a capacitance and resistance, and all the other components are represented by a resistor. So the equivalent circuit will be as shown in the figure. And this is the equivalent representation of an electrode-electrolyte tissue interface, which also shows the other layers down the skin. So let us now look into the electrolyte tissue interface. We know that at the boundary between the electrolyte and tissue, it is actually separated by two aqueous ionic solutions of different concentrations, separated by a selective semi-permeable membrane, and so an electric potential exists across this membrane, which is given by Nernst equation. Accordingly, the potential that exists across the semi-permeable membrane is given as E is equal to minus RT divided by NF, natural log of C1 F1 divided by C2 F2, where R is the gas constant, which is equal to 8.315 into 10 raised to 7 ergs per mole per degree Kelvin. T is the absolute temperature in degrees Kelvin. N is the valency of the ion. F is the Faraday constant, which is given as 96,500 coulombs. C1, C2 is the concentration of the ions on both the sides of the membrane, while F1, F2 is the respective activity coefficients of the ions on either sides of the membrane. So that completes the whole representation of the electrode skin interface. Now let us look into the various types of biopotentials that are biopotential electrodes that are possible in acquiring a biopotential due to even though there are different types of bioelectric signals available from the human body, the most important among them are the electrocardiogram of the heart, electroencephalogram of the brain, and electromyogram of the muscles. The earliest bioelectric potential measurements used immersion electrodes, which were simply buckets of saline solution into which the subject placed his hands and feet, one bucket for each extremity. As might be expected, this type of arrangement presented many difficulties, such as restricted position of the subject and danger of electrolyte spillage. Now, this arrangement was actually made by the father of electrocardiography, Professor William Einthoven. Now, great improvements were made over the immersion electrodes with the development of plate electrodes. Now, based on the type of biopotentials acquired, there are three types of electrodes available. The first one is the microelectrodes, where the acquisition can be from within a single cell. The second type of electrodes are the skin surface electrodes, which are basically used for EEG, ECG, EMG potentials from the surface of the skin. And the third type of electrodes are the needle electrodes, which are used to penetrate the skin to record the signal. So the microelectrodes are generally of two types, metal and micropipet. Metal microelectrodes are formed by electrolytically etching the tip of a fine tungsten or stainless steel wire to the desired state. The wire is then coated almost to the tip with an insulating material. Some electrolytic processing are also performed on the tip to lower the impedance. The metal-ion interface takes place where the metal tip contacts the electrolytes either inside or outside the cell. So these are the different pictures of microelectrodes. As seen from the picture, the structures show metal-filled glass micropipet and glass micropipet coated with metal film. Second type of biopotential electrodes are the body surface biopotential electrodes. So these type of electrodes are used for the application to the limbs. They are available in the disposable type of foam pad electrodes, which are often used for the capturing of electrocardiograph signal. So the different types of disposable electrodes. However, the surface electrodes have difficulty in using sometimes when there is always a possibility of the displacement from the actual position of acquisition. So under such cases, suction electrodes can be used, which is associated with a rubber bulb, and they can be pumped in such a way that the electrodes will never move or will not move from its place. So it can be held tight on the surface. Another development was the floating electrodes. The principle of this electrode is to practically eliminate movement artifact by avoiding any direct contact of the metal with the skin. So here, the only conductive path between the metal and the skin is the electrolyte paste or jelly, which forms an electrolyte bridge. We also have the flexible electrodes available by various manufacturers. They are mainly used for newborn infants, and it's also compatible with X-rays. The third type of electrodes is the needle electrodes or wire electrodes. Now, needle electrodes are used to penetrate the skin to record EEG potentials from a local region of the brain or EMG potentials from a specific group of muscles. They may be very slightly penetrated under the skin. However, in animal studies, they may be penetrated deep down to acquire the necessary signals. And this slide shows the picture of a few standard electrodes. With that, we conclude this presentation. Thank you.